collecting these samples, and everybody works together as a team. So, and then of course, I'll miss being fed. When I go home, I have to fix my own food and wash the dishes and all that sort of thing. So it's nice being taken care of out here.
Aaron? Oh.
I think kind of building on what Dean said, or Dane said, it's really nice, the teamwork aspect of being at sea. Like when we brought up the samples at the end of the last dive, and you have 6 people in the lab, 2 different sides, 2 different teams, just getting stuff done, helping hands basically everywhere, and people just really open to helping you get what you need to get done. And it's a really great atmosphere.
It is.
Very healthy.
It is. And, you know, I mean, the people that I've encountered on the OET expeditions are always really positive, and they really love their jobs, and they work hard at their jobs, and they're there to accomplish the goals of the mission. And I mean, it's really professional. It's a pretty unique environment, I think.
Mike?
He's not listening.
No, I think they're busy with tech stuff.
What about you, Scott?
Someone follow me?
My gosh, you know, when I went to the SCF training, well, so the first time I went on an ocean research vessel, I had this great sense of sort of being where I had always wanted to be. I know it sounds trite, but I'm one of those people that was inspired to ocean exploration by the undersea world of Jacques Cousteau. And coming aboard Nautilus and meeting the people, everybody shares that same passion for this and for exploration. And nowhere else have I ever felt such a sense of, like I said, sort of being home with like-minded people. And yeah, I definitely miss that. I mean, I love my job. You know, I have a great family and all that. And I know that my wife is probably going to type in right now. But this is, you know, and this is that thing. Just that great sense of being where you've always wanted to be, and like I said, with like-minded people. So, yes, I'm going to miss all of this.
Well, the discovery. I mean, for me, science is about the sense of discovery. Finding new things, finding out how things work together. How do you explain that pattern you saw in the data? It's invigorating.
Hey folks, as we're approaching bottom right now, so we might be chattering some more over this line just as we get ourselves all set up.
Sure.
Just because we're about 80 meters off bottom.
All right.
Okay, and I wanted to, there is a question here about what our objectives are. I don't know, Jennifer or Annie, do you want to share with our viewers what we're about to embark upon?
I wasn't going to believe that one.
Well, the objectives of this dive, as we've been saying, is to go see some new things. So we're descending, we're getting close to the bottom, and around the outside edges of the Davidson Seamount, and areas that we've never been able to explore and look before. We'll be looking at the geology, so the rock formations and the associated animals, particularly still keeping our eyes out for aggregations of octopus, unusual conditions where water may be venting out of the seafloor and causing a shimmer. Also looking for aggregations of sponges and corals, or other species that would be on our list of species that are new or being studied, and we've been asked for collections of those. So those are some of the things we'll be looking for. Dane is going to be helping us collect water and sediment samples to look at contaminants.
Tell Mark we're going to stop now in a minute.
And lastly, some other water samples for genetic and genomic analysis.
And we're also going to be looking at environmental variables. And so we have these special probes on board that can look at temperature and oxygen. And then the Hercules itself is equipped with a CTD that measures the conductivity in the water and the salinity. And so we get to really try to get a picture of the types of water conditions that these organisms are living in.
Great, thank
and then
you.
the samples we collect, those will get sent out for further analysis of the geochemistry.
Great.
And we're expecting a 30-hour dive?
We're expecting, I think, a weather-controlled dive.
Yes.
Yeah, but around that.
But around 30 hours.
Yep.
And that's the amazing thing about this operation is you can be on the bottom. You can have your eyes on the bottom of the ocean for 30 hours straight.
How far are we off the bottom?
40 meters.
40 meters off the bottom, but we're getting the vehicles oriented here, and then we've got the deck crew's got the cable looper on the cable, so we'll have to wait till they get that off before we go down.
Awesome.
So how many people, I know we have the ROV team and the navigator and the data logger and the video engineer in here, so that's 5 of you. And then how many deck crew work on this operation that are related to the Nautilus itself, that aren't like visiting scientists and communication
Well, we only have
interns?
one dedicated Mark runs the deck.
Okay.
And everybody else out there has another role.
Okay.
So when we mustered for our safety exercise the other day, there were 41 of us. So 41 of ROV science crew, and then there's the ship's crew.
Which is 17.
17.
No, that's included in the
No.
Yeah.
No, they counted
30,
through us.
31.
Was it 31?
31.
Okay,
Yeah.
31.
47 total on
And
it.
then-
Okay,
Yeah,
Sorry?
got it.
got it. Yep.
Yeah, I'm holding right now at this depth.
And it's actually kind of crazy because everyone's on different schedules. You can go an entire week
Right.
and not
Yeah, I
see
can do that.
Yes.
all 47 people.
Right.
I was just going to wait until, but yeah, no
And the
point.
boat's only, what, 200
And
That's
11
meters or feet?
feet.
211 feet.
No, is that right?
I thought it was 280 something.
And this is a site that
No.
no one,
Magda,
but
how long's
no
the boat?
one, at least
Right.
that we know, perhaps Captain
Okay, I
Nemo,
was right.
211 feet.
I'm sorry, how far? How long?
no one has been at this site before.
Correct.
211 feet.
211 feet is Nautilus.
Just less than the size of a
Yes,
football
when
field.
we see the bottom, we will be the first people to see this little section of the seafloor ever in the
Are we there
history
yet?
of the world. And everyone watching with us will have that same thrill. First time ever seeing it.
I think we're going to see sediment. I think that's what we're going to come down on.
If you see
My
that,
guess.
that green V in the screen, those are our lasers. The points are 10 centimeters apart. So we use that as a scale to measure things as we're going along on our dive. Oh, sorry, I just thought I saw the bottom.
One thing I won't miss when I get back is having to translate in my head between UTC and Pacific Daylight Time.
Is home for you California?
It is.
Okay. I have to go an additional translation back to
Oh,
East Coast.
Right, okay, yes.
I've been avoiding that by just peeking at my phone.
Yeah, I do that too.
I should have a watch on, but I don't.
Oh, I carry my watch in my pocket.
With your phone?
It's my phone.
Oh.
We're at 219 visitors online right now.
Wow.
All right.
Ecuador has joined us.
Hello,
Austria,
Ecuador.
Argentina.
Buenos dias.
Can you also see if there's anyone watching on YouTube? I think
Let's
were able to check
see
that
here.
somewhere.
You know, I'm trying to learn my way around this thing. Ooh, but one of the things that I can see is that of the 220 visitors online right now, 179 are returning.
Wow.
Oh, wow.
Yeah. Here's a Twitter search.
Hey, is that it? No.
In ecology, we call that site fidelity.
Yes. I cannot tell on, sources? No.
Oh,
No, I can't tell, but you know, yesterday somebody buzzed in, and I think they said that there were a significant number of people.
Oh, okay.
Oh, right here,
That's
we got
what
the
it was.
answer. 39 are on YouTube. Thank you.
Okay.
Oh, okay.
Thank you, Nautilus Live Nation. It's nearly squishy creature time, says somebody. Yes, it is.
Nearly squishy
Bring
creatures.
on the squishies.
Yeah.
And there was squishies, and it was good.
We have been seeing some squishies in the blue water already. They go by awfully quickly, so we don't get a chance to look at them as closely as those that sit still for us on the seafloor.
Should be a game every time we quote a Finding Nemo.
Yes.
030, Roger.
It's happened a lot. We've quoted Finding Nemo a lot.
What,
on this
just,
cruise.
I
The
missed
squishy,
it.
That's from Finding Nemo.
I know I said journey,
Oh.
but get to the bottom, get to the bottom. 109 are watching now.
Wow, on YouTube?
Oh, yeah.
That jumped up real fast.
Oh, yeah, I think there is, all of the Ethernet is a Twitter.
Well,
that we are just about to hit
Oh,
the
yeah.
bottom of the ocean for which
Oh,
no
is that
one
it?
has
Oh.
gone before.
I
We
think
have-
we're seeing
There
spots.
it is. It is
There it
mud
is.
bottom.
And it's sediment. Woohoo!
Yep, Annie won.
No one else guessed.
Well, okay.
It's going to be mud or rock.
We
And what
only
is
had
that? Is
one
this a
entry,
Cuskill
but
we have
Annie won.
to
That might-
welcome us?
Yeah, might be. Or it might be a grenadier. We'll have to see when we get closer.
Or we won't. A fish greeted us.
There's a fish greeting us.
Ooh, now,
Hmm?
no, that's more
I'm getting
texture
sedimental.
in the mud. Yep, sedimental.
Is that a frog?
That's
Uh,
right.
Frogfish?
We get 30 minutes of
No.
bottom.
Woohoo! So this is a predetermined transect we're going to be following from point to point,
Well,
right?
it's not going to be a transect. We're going to be in exploration mode, going generally from one waypoint to the next.
Okay.
So we've set a series of points that are guiding us
So
along.
like breadcrumbs.
Yes, but we are not required to go in a straight linear fashion between them. We are allowed to meander a bit, particularly if we see things that draw us in one direction or the other, or if the, you know, we see, you know, something on the radar that looks good or something like that. Sonar, I mean, that looks good. We might want to go over and see what it is.
Like a,
Very
is that an
soft
acorn
stuff.
worm?
Yeah, we're already
behind-
seeing some sea urchins and some corals. And
Another
here,
one of those fish.
And here comes a grenadier.
Yeah, we're going to catch you.
Ah, we're just kidding.
Do a white balance check.
Yeah, we're getting you set up right now.
Okay, thanks.
Okay, zoom in there.
Right now, our video engineering and our ROV team are working together to make sure that our
You know, bump up if
pictures
you
look
don't wanna do
okay.
that.
Yeah. coming
come
through.
on. That looks better. There you go.
So that all these photos and videos that we capture look their best.
All right, go ahead.
Okay, we'll go black just a moment.
You.
Oh, I love you guys out there, Nautilus Live Nation. Yay, squishies. Torch is out, time for science.
Okay, white balance complete.
Roger.
Look at right in front of us, this weird little divot. I wonder, is that where we landed? Did we touch down there?
I don't think we've touched.
No, we haven't touched. It's our thrusters.
Oh, that's
Just
two of
came
them.
close.
Someone wanted to know, does the cable from
Alright.
Argus to Hercules drag on
Yeah.
the bottom?
Okay, Mel, if you had to jog through
Don't say
the
that.
jog through the autos.
No,
I believe
that
the
would
cable
not be good.
has floats on it
Okay.
so that it does
There's
not.
a
Yes.
burrowing anemone.
No, the cable isn't just a cable, right? The cable is like a very, very technologically advanced communicator,
Yeah,
right?
it's
Yep.
a
Fiber optic
tether
cable.
or umbilical. Actually, you can look at videos on the internet of how they make these umbilicals, and it is pretty amazing. It's
Yeah.
giant spindles or spools that put all the different components in there because they've got wires, like copper wires for electricity. They've got fiber optics in there. They've got like Spectra, Kevlar type fibers for strength. Got inner jackets, outer jackets, like a double helix of wire wound on the outside. It's pretty neat machine. Pretty cool video to watch.
I looked out and have actually a piece of the tether because I was on a cruise where they had to replace it. And so they gave some of us who wanted it a piece of the tether, and then they even stripped the yellow
Sheath
protective
off.
sheath off. So I have a piece that has the yellow sheath on and then a piece that has it off. And
Is it heavy?
yeah, it's heavy. And so you can really look inside. And there's three, I think there's three parts inside that are all the same for redundancy that have the if I'm correct, that have the fiber optic and the copper. They have three of that so that you just aren't relying on one.
You can imagine
Redundant systems.
having to move all of that cable
What's it doing?
sitting 3,000 meters through the water column as we move these vehicles, and that's one of the reasons we
Oh,
have to
wow.
go so.
Slowly.
Yes.
Yep.
Now I don't
fire
have
it.
any of the
Just undo the button or
tether that goes from Argus to the
or,
ship.
Right.
I
cycle
only
the
have the
DVL.
yellow
What the hell?
tether that goes between Herc and Argus. Yeah. Though I did see them
might
cut
need to
through
restart the GUI or something.
the
I'd
metal
rather
one, and
just
I have
leave it
photos
alone.
of that if you
Shouldn't
want
need
to see it. It's pretty cool.
Stupid.
Are we touching down? Okay.
Just getting our bearings here.
What? Oh, I do. Yes, that's definitely. Okay. So we're still getting situated here.
6.1.
You can see we've come down in an area with a soft sediment that's built basically up over time by the marine snow that's slowly drifting down and settling on the seafloor.
Never sit down in that unless you need to. You can hold still and get a DVL fix the same as sitting down. You just make that stuff's so soft.
Dane,
just
when are
make a
you
mess.
going to want to start the pump?
Once we start transiting,
Okay.
but actually, I'll want to have eyes on the exhaust when they turn it on.
Okay.
Now, yesterday your sample didn't go so well.
It, yeah, the intake hose got crimped by the manipulator.
Will you still be able to use the amount of sample that you did get? Will they still get an
Yes,
analysis out
I
of that?
just won't know the volume of water that we pump through.
Okay, so.
So, because it changed through time, but it will be useful in looking at the relative amounts of the different kinds of compounds in that sample and
Okay.
comparing it to what we get with the quantitative sample. Yeah.
Good.
All right, Erin, go ahead.
Can you let me know if you said? Okay, thanks.
I'm ready for you if you want. Yeah, you can go ahead and keep an eye on those. So that change.
I should have written a message on the squeezer tops.
Hi, Mom.
Happy birthday,
Yeah, right.
John.
Okay. Where's the first place we're going?
Okay, well before we go anywhere in particular, we wanted to make sure that we didn't forget to fire a Niskin for background. So
Right.
I thought we'd do that as soon as we got to the seafloor.
Okay.
So what Jennifer is just referring to is we're going to use this
There's
special
no good hits
tube
there, is
that
there?
Oh, we don't want to fire it when we have sediment.
So no good hits,
Yeah.
is there?
So let's
Okay.
wait until we get clear of that.
Okay, just let them get their DVL fixed and then we'll push off
Okay.
somewhere.
it's a special tube that closes shut really quickly when the ROV arm triggers its enclosure, and the water that is sitting in the tube at that specific depth gets captured, and then it's not able to leave the sample tube. And so when we carry it back to the surface, we have a solid bottom water sample that hasn't been mixed with any other depth water.
Yeah,
USBL
that
is just
might-
terrible, is it?
And we're calling it background because we're not taking it by anything.
Data,
in
I'm going to be
particular.
resetting the DVL in about 2 seconds.
Just supposed to be a
Thank you.
representation of
Okay.
Yeah, go ahead.
the normal homogeneous bottom water.
Further on in this dive, if we see high densities of corals and sponges, we'll also then take a Niskin sample, but in that case, we're sampling water near those organisms.
A little louder.
Good.
Yep, thank you.
Are we getting
Yeah,
another
sure.
eDNA Niskin on this one?
If we
you
fire
catch
the
all that, Jess?
Niskins near corals
Yeah,
and
so I'll
sponges,
get out of
we'll
that
look for
cloud.
eDNA.
Okay.
Yep.
That's such a new thing to me, this
Yeah,
eDNA.
I know. It's exciting.
Yes, this is happening now.
Okay, so now are we transiting towards our first waypoint?
We're
Nope.
going to get you guys a
Just
background
pushing
water.
out of that cloud there,
Okay.
get you your Niskin.
Okay, great.
All right. All right, I'm about 3 meters off bottom here for you.
Camera back in.
Do we know what the
Yes,
pressure is at
camera's
this
racked
step?
in.
Porch
Your
light.
porch light.
Can we do number 7?
Roger.
Thank you.
I have eyes on the camera.
So when they pull this little red tab, they're going to be triggering the Niskin shut, and that's going to capture the water at this depth.
Are the Niskin samples sent to a specific lab?
You got eyes on it?
Yep.
Hold on one second.
Fired.
So the water has been requested by a researcher who's looking at environmental DNA from corals and sponge aggregations in the deep sea. So if we fire the Niskin near those, that's where they will be going, and this background one will help
All
be something to compare against that sample that was taken near corals and sponges. Right.
Now that was sample 19.
Do corals and sponges, do they like
Aaron,
shed
did you
skin
drop
cells and
did
stuff?
you have to drop a point on that
What they're
one?
looking
do,
for in
thank
that
you.
water column, or
Tissue cells, yeah, I would think.
Got
I
it.
don't know if they'd be called
Or not
skin
skin,
cells
but
per
tissue.
se, but
Data, what
Also-
sample?
19.
they do expel, waste, and that can also contain DNA. Okay, so we've got our Niskin. Dane, when do you want to
Yeah.
start the pump?
Yeah, so if I can get the bubble cam on my exhaust port there.
Oh.
I'm not seeing it.
there it is.
Ah, yes, there it is. Okay, so Jessica, you ready to start it?
Sure thing.
Okay, let's go.
All right, PCB pump on.
2238.
Pump's on.
All right.
Yep, okay.
Looking good?
Yep.
All right, so pump is on.
Oh,
So we're going to
yeah.
want to be careful
You want that again?
when we,
Sure, thank you.
if we kick up sediment,
Yeah.
that we turn it off.
Right.
And pilots, if you can please let us know if you turn it off so that we
Will
can
do.
note that. So we're keeping track of cumulative time that's running, so we can get 10 hours total time.
Sure thing.
Thank you. Okay.
We are in bottom water temperatures of about 1 degrees C, And we have an oxygen concentration that is 119.7 micromoles.
Do we know what the pressure is?
Sorry, so where are we?
Let's say Argus is there. We're headed up there. Looks like bearing about 033, 60 to 70 meters.
Okay, 033, roger.
033.
Aaron, we're heading to drop point dive 2 on the map, is that correct?
Yeah, is that what you want?
That sounds good. Thank you.
All right. We ready to go?
Yep, sounds good.
All right, I'm gonna step it.
Let the journey begin.
Bridge nav.
And we're off.
Oh, that's not gonna work.
Or is this the
Westward
destination?
ho.
Again, our start point was predetermined using the mapping information available to us. We didn't set it knowing that there'd be something particularly interesting in that location, but we're just kind of setting up a track that we thought would bring us potentially near some interesting changes in the seafloor that might have associated interesting biology.
If you're just joining in now, we are on our final dive, on our final cruise of the 2019 season. This is dive H1796. We are on Davidson Seamount
Chess, I anticipate we'll be going
in
pretty sharply down
Monterey
slope at certain
Bay
points.
National Marine Sanctuary.
It might be the
Sounds good.
slope that's playing havoc with the USBL.
The southeast apron.
Yeah.
It's not very...
And I just found out that that dive number you called out, if you reduce it by 1,000, you know which number dive Hercules is on. So this is Hercules dive 796.
Wow.
I just got Nautilus bingo.
That would be fun. We should make up some sort of a
Game.
yeah,
I'll give
deep
you my
sea
nickel on
biology
the way
bingo.
out, Scott.
Deep sea science bingo. And if you say Niskin, you know, people can mark it off. If you say persistent organic pollutants,
Yes.
mark it off.
Bazingo.
Bazingo?
Bazingo Sea Star.
Oh,
Bingo.
Brazingid.
Brazingid.
Brazingid. Yeah.
Brazingid Sea Star. Gotcha.
B-R-I-N. No, B-R-I-S-Z.
B-R-I-Z.
Isn't it S?
I think it's S.
B-R-I-S.
Oh, yeah,
I-N-G-I-D.
right.
I was mixing my science with my pop culture.
I
And
was thinking, you're thinking of Bazinga.
yes, that would be the TV show.
Aaron, the green trail on High Pack, that's supposed to be Argus?
That is supposed to be
That's correct?
Argus. Yep. Green is Argus.
Big Bang.
Brighter red's HercuSBL. Darker red is TBL.
Yeah. Looks like the sonars don't agree with that slope.
Okay, I'm getting a lot of pressure from the interwebs here. People want to know what the pressure is at
It should be
10,000
downslope, right?
at
So
our
slope's
depth.
They
behind
can
you?
calculate
That
that.
makes sense, right?
So we're
Oh,
at
I'm
a
looking
depth
at it
of
wrong. Okay. Well, then that makes
3,200
sense.
meters, and for every 10 meters of water depth, you get 1 atmosphere of pressure.
Ah, okay.
You got a feel for it?
Yep, yep,
Okay,
yep.
So that's the challenge.
And
What's-
that's
what's the
colored by
pressure we're
slope
at?
too.
Right. Oh, okay.
The backside of that slope, yeah.
So 3,200 times atmospheric pressure.
Now, is it possible for us to get just a little bit closer so we can see the seafloor while we're transiting, or is that going to slow
Or
us?
320 times.
Yeah,
Right.
sure.
Oh, great.
Here we go. We are going down slope, so we're going to have relatively limited visibility.
Okay.
I don't want to stir up sediment for that pump, but
Yep, thank
Sure. But just in case
giving you, a tug.
there's something
Oh, sorry.
cool.
Ooh. Spicy.
All right, all the way forward. Yeah, as
I've
you can
just
see, the
been
slope
informed.
doesn't work in our favor here, but
Okay.
We can kind of watch an Argus.
You can
Hmm.
you can stay closer and
I can lateral it a little bit.
corner a little bit so
before
they
I-
can see something, and then there's not so much. I'm not so much further upslope than you.
That's true.
I've just been informed Nautilus bingo already exists. It has for a while. It's on the
Just
website.
watch it.
Yeah.
Well, that was a
Thank
good
you,
idea.
Nautilus Live Nation.
What was that? I could barely hear you.
Nautilus bingo already exists.
Oh, really?
Hey,
Yeah.
Dane.
Yeah.
Dane,
It can be found on the
we have a
website.
little cloud behind us. Should I
That's
secure your pump? Or is that all right?
No, that's okay. Yeah.
All right.
I mean, we still have a clear view of Nautilus, of Hercules, and so it hasn't.
You just don't want it
overtaken.
coming up along the sides, is that right?
Yeah, I don't want it coming over the top. Right.
And the intake is on the top?
It is.
Okay.
What direction? Are we just going over to this drop point here?
Yeah.
Okay, so
And it's a suggestion. It's not like it has
right.
to
Like
be right
where's
on there.
where do we go from there? Because
Yeah.
do you just want to do that?
So we'll have a next waypoint, and I think we're just going to start out, go into the next waypoint after we're oriented here.
Roger. Hey, video, are you full wide on Argus?
It looks like it. No.
So, Jen, is this waypoint important? I thought it was from the dive plan, but if not, we can move over to
It's
the
a
other.
suggested starting point.
Okay.
We don't have to be right on that spot.
Okay.
Where's the next one though? Is it the one to the east or north?
Up
Looks
there to the
north.
north. It's 700 meters north. Jeez.
But I think we would, instead of going as, you know, the crow flies, we'd want to kind of more stay in this, along these contour lines.
Sure,
Yeah.
you want to be
Yeah.
along that high slope area.
Yep.
Mm-hmm.
Yeah, 700 meters straight up, but slow going. Yeah, nearly directly north, but we'll go along this.
Okay.
Yeah. So if you want to go along the slope, is actually. Wow. You know what I mean, Aaron? You're just going to do a few different directions, right, to stay on the.
Yeah.
Like as the crow flies, goes right up over the steep part.
I mean, we'll have to cut it in a bit more. Yeah.
Yeah. Yeah. So I don't see any reason to go into that start point there. It only takes us away from it.
Okay. Well, this time we're still moving you into like the higher slope area.
Yeah.
So just keep it going a little bit longer if that's okay. And then start moving to the northwest.
Yeah.
All
Yeah,
right,
sure.
Bridge nav.
There are 30 different
I
bingo
have
cards
another
of Nautilus.
20 meter
bingo.
step, same bearing.
That's fun.
Thank you.
Let's just go that way.
Yeah.
There's a Nautilus knit along.
I think, I mean, if we
Yeah,
we
I
start
think just
going
start
that way,
going
it'd be
that way.
best.
It's all going to drag out behind the ship.
As long as that's okay with science.
Yeah. I think, right?
Yes.
Head toward the.
Head toward the steeper.
Yep.
Yep, head toward the steeper section. Yep.
Right?
Right.
We got it.
The expected length of
we
this
saw
dive
the bottom.
is
We saw the bottom.
32 hours. However, it is also weather dependent, but our goal or expected dive length is 32 hours.
I think I'm debating it if we're either here or here. I can't tell. Sorry.
Again, if you're just joining us, we're in Monterey Bay, National Marine Sanctuary over the Davidson Seamount. We're at the southeast apron,
Like come
an
up slope
area
a
that
little
was
bit,
only
Jess.
mapped 2 years
Yeah,
ago.
sure. I'm just trying to see if I can get one more hit.
And
I think once we
you
get some
are all
movement going,
seeing
that
it
should
for the
straighten
very
itself
first
out unless
time.
there's something else wrong with it. Like,
That's
I
true.
just don't think it likes where it is.
That's fair.
It would be sweet if these had like a little pocket protector. Yeah. Pen slots.
And that dive time includes deployment and recovery.
Oh, it says.
Oh, that right?
So we deployed shortly after 1 today.
Set line.
What is that dark?
It's what?
It looks like a cliff edge, like we're just coming around the.
Uh-huh.
That's the shadow that we
Yep,
can't
that's
see.
right. Okay.
We're bringing you some fun stuff, looks like.
It's just a slope.
Oh.
Is it?
Yeah, it's just the relief.
I'm gonna switch over to my replacement Amanda because she's standing behind me, literally identifying things in the water column. And I think it's more advantageous for you guys
Sorry,
to hear
Mike.
her and me to be able to hear her. So it's been a good dive. And here's Amanda. Oh, yeah.
Yeah. Yeah, definitely. That's fair. Actually, maybe we should increase the gain. Yeah. Aaron, what was the bearing on that ship move that you just did? Was it due north?
No, 348.
348. Roger.
Just to get my bearings, are we looking downslope right now?
Correct.
Okay. So now we're swinging around.
Just getting everything situated here. So we're going to be looking up at the slope mostly and then going along it just as we were yesterday. If that works for you.
Sorry, could you repeat that?
Just turning the vehicles around now instead of looking downslope like you see Herc now. It looks upslope and ahead.
Yes.
And then we'll go on like that, just
That
like we
sounds
were
great.
yesterday. And then we just need to change the bearing of the line a few times as it proceeds northish just to stay on the slope.
Sounds good. And then we let us know if you see there's some hard bottom features that are nearby that we might want to check out from the is it Herc or Argus
something
that
that we
has
see in
the
the sonars.
yeah, the
Yeah,
sonars. Yeah,
not
if you
see-
see interesting sonar targets, we definitely want to know.
For sure, yeah. Not
Yeah.
seeing anything there now, but
Okay. The pump is on.
Yep.
Yep, and we're going to want to make sure that if any of the sediment starts to billow up and get up the sides of Herc, we want to turn the pump off and note that, and then turn it back on when the water clears.
And so data were not, I think there was some confusion about logging the on-off as separate samples.
Yeah, it's one sample.
It's one sample.
Yes,
Yep.
yeah, thank you for
Good
that.
job.
Yeah.
Mhm.
And
So
Another step, please.
in addition to, so there we've got a Grenadier in
Yep.
view.
Mhm. Yeah, the Mesotech sonar on Hercules is showing basically soft sediment, soft bottom around it.
A lot of things on this sediment. Looks like on the left-hand side, that string, green string, my guess from this distance is a spoonworm, because we were seeing a lot of those at the other site.
One thing else that we did see, though, would be seagrass, too.
Really?
We did see some seagrass.
Oh, well, that doesn't grow here.
Yeah.
No, I
That's
know. And we did see some kelp, too. A kelp
Oh.
Yeah,
stipe
we
and
did,
some kelp
right.
fronds at one point. So
See, there's more coarse material up there.
Can we zoom on that, please?
Yeah.
You
Go
guys
ahead,
are on
video.
it. Who is it?
I think it's going to be
That's it.
kelp.
I think
It
it's
is.
it is
It's
kelp.
kelp.
Oh, I
That
win, I win
is
bingo.
amazing.
Guess what, Jen? There's cookies.
There's cookies? I just
Yeah.
want a cookie.
She just want a
Bing,
cookie.
bing.
Well, there's a whole
Alright.
plate of them up in the galley, though.
Hey, no. Extra cookie.
It feels better if I think I won it.
That's true.
Exploration cookies. Exploration cookies.
That's
of the
right.
So I'm going to go quiet for a little bit while I fill in science on what we've completed and the plan moving forward here.
Bye, Jen.
Thanks, guys. Bye, Jen. Bye, Annie. Yeah, so we're just about to do a shift change here. We are in Monterey Bay National Marine Sanctuary on Davidson Seamount. We're exploring the southeast apron. We're going to be doing so for the next 29 or so hours, weather permitting. We are at a depth of about 3,300 meters. And there is Science Communication Fellow Danny, who will be taking over for me now, which means I have to give up my seat.
Oh.
Can I do a DVL reset?
But I will be watching live
Okay.
in the lounge
Stayed about
aboard
there.
Nautilus as this never
Zeta, are you
before
on
seen
comms?
seafloor continues to open up for us.
Taylor,
All right.
can you hear me?
Bye, Scott.
Goodbye, guys.
Yes, I can.
Okay, I'm about to do a DVL reset, so stand by. You want to log that?
You're going to do a what?
A DVL reset.
DBL? Okay.
Yeah.
All right.
Morning. Or, yes.
Okay.
All right. Reset.
Right.
Good. Morning.
Yeah, that's a fact of these 4-on, 8-off schedules. Whenever you see somebody coming in to start their time on, it's always good morning. Yeah. See you, Scott. Okay.
Hello, Danny. Are you on SCF? No, okay. Huh?
Hear you on there.
I'm on SCF. Hello?
Yep, we hear you.
All right. We're doing a little bit of a shift change here, I think, as everybody is getting settled. Thank you for tuning into Nautilus Live. This is Danny. I am your science communicator, and we are here to answer any of your questions that you may have about the deep sea. No promises for answers, because we don't have all of them, but we love to hear what you're thinking, and we'd love to start a discussion. So please come hang out with us, let us know what you're thinking, and thank you so much for watching.
Oh, and they want a reminder to take the temperature and DO before using a squeeze sample in that exact area. I have that note here.
Hi, Jessica. Thank you.
That looks like a squat lobster there towards the center. Long distance.
Bye, Jen. Amanda, look at that sort of...
That dark thing that the lasers
No,
are
the
on?
escarpment.
Yeah. Can we
So-
zoom first on the little squiggle that's between the lasers?
Sure.
Now it's to the right of the lasers.
Good afternoon. 4 to 8.
Good afternoon.
Hello, hello.
This is exciting. Ah, it was a
Dan, can we
sea urchin.
move to the right a bit? Starboard?
Right. Sure.
There was a feature
So uphill,
request.
That dark thing there.
uphill is to my southwest. Which way are we going?
We're going northeast.
Ah, perfect.
Let's see.
That looks like the sediment is clay-like. Or it's-
Yeah.
Yeah.
It's down at the bottom of the screen now. Just went off
Really
screen.
quite sticky.
Coming wide.
Roger. North.
Okay. We'll never know.
Moving
Never
north.
know.
That's all right. We'll see more squiggles. Well, hello.
Well, hello,
We
Amanda.
have
Welcome back.
just had a swap. So we have the 4 to 8 watch led by Erica Burton.
Erica Burton from Monterey Bay National Marine Sanctuary. Good evening.
And hi, my name's Amanda Kahn. I'm from Moss Landing Marine Laboratories.
And I'm Taylor Ann Smith. I am the ocean science intern and data logger.
And I am still Dane Harden.
I'm Miles Saunders. I'm a navigator, and I live on the boat.
Whoa.
I'm Dan, your taxi driver for today.
Antonella, Argus pilot.
And I'm Wayne, your video engineer.
Hello, I am Danny, your Science Communication Fellow. It is great to have you guys joining us. I see all sorts of fantastic questions coming in. To cover the first important point, they are chocolate chip cookies.
Of which I have 3 in
You
my
had
pocket.
3 in your pocket. I only have 1, but 3. You are prepared for the shift.
I am prepared for shit. Well, it has to get me till about, what, 5:45 when they relieve us for dinner?
Yes.
So I got 1 cookie for each 15 minutes.
I think you're well set.
I think I'm okay.
All right, but we have
Oh,
plenty
hello,
of
Sea Star.
other questions.
That looks like a Pteraster sea star, I believe.
It does,
The
doesn't
squishy
it?
cushion ones.
Yes.
I love the squishy cushion ones. I just want to hug them. I know they're not as soft as they look, but I just want to hug them.
They're pretty soft, but they're pretty voracious too. I get kind of cranky when I see them eating a sponge. I'm like, oh man, no.
Pick on somebody your
They
own
try to
size.
eat everything, really. They do try to eat everything.
Yes.
We were tuned in last week, and there was a sea star sitting on top of a fish. Or was that our cruise?
I don't recall.
I've been watching a lot of Nautilus dives this year. But yeah, the fish was just sitting there on the bottom, and there was a sea star on it.
I did not see that.
Yeah.
All right, so for everyone that is watching, we are off, I want to say the southeast side. Is that correct?
Yes.
Southwest? Southeast?
Southeast
All right,
side.
we are off the southeast side of Davidson Seamount in the Monterey Bay National Marine Sanctuary. So we are exploring an area that has never been explored. So you guys are joining us in seeing an area that has never been seen before. So we have no idea what we're going to find on this adventure. I know you guys are a little sad that we haven't found any more squishies yet, but don't worry. I'm pretty sure they're down here. We'll find some more. But while we are looking, I have a fantastic question of how is a sanctuary determined? Scientists, can you help us out with this?
How is a sanctuary determined? You mean should there or should there not be a sanctuary, or how they come about?
Yeah, how does it come about? What are the different factors? And yeah, should it not be? What parts are, what parts aren't?
Yeah, so in the National Marine Sanctuary System, we have 14 sanctuaries and a monument, and they're all sort of designated for different reasons. We have ones that are dedicated to shipwrecks. One is the first sanctuary was the Monitor on the East Coast. We also have another shipwreck sanctuary up at Thunder Bay. But the Monterey Bay National Marine Sanctuary was designated to stop oil drilling from occurring off our coast. It was happening in Southern California, and there were some oil spills, and there was a grassroots efforts in our area back in the 80s and the 90s to get those not to have those off our coast. And the grassroots effort said, how can we do that? And through the National Marine Sanctuaries Act, there's a regulation for no bottom disturbance and no oil and gas extraction. And so in 1992, through grassroots effort, the sanctuary was born. And we, the Davidson Seamount came later in 2008 when we had done some preliminary cruise work and surveys to see what was on this big bump off our coast. And it turns out that the tippy top of the sanctuary of the Davidson Seamount were lots of large corals and sponges the size of your sofa and your cars, and it was pretty spectacular. And those structures actually make habitat for other living organisms, and many people thought, fishermen, fisheries regulators, the conservation community, ecologists thought this is a special place. We need to protect this for future generations. And at the time, the Monterey Bay Sanctuary was undergoing management plan review, and it was sort of a natural time to say, hey, let's try and get this part of the encompassed into our sanctuary. And that's how this one came about. I don't know if I answered your question, but it was sort of specific to Monterey Bay Sanctuary.
I think you definitely did. That gives us a good idea of how these are determined and what was sort of the starting point for the current one that we're in. So I think that's a great idea, or a great way to answer that question. My next question is, if you could walk on that sediment, you know, without exploding, freezing, and compacting to death, would you just sink? How deep is it? So what are we kind of looking at with our bottom sediment here? Is it soft and squishy? Can we, in theory, walk on it? Would we be able to sink? What kind of geological origins do we have?
Yeah, so the seamount itself is of volcanic origin, and typically up around the seamount has a thin veneer of sediment dusting. But we can probably dig down about a foot in some places, maybe a meter in some places, but for the most part it is a thin veneer of sediment on this volcanic rock. Now we're down at the deeper parts of the sanctuary, so I expect this to be a little more squishy, a little more sediment as we're approaching the muddy seafloor. It's not part of the volcanic structure.
So if you could stand on it, you'd probably sink in about 6 inches.
Probably here,
and
yeah.
And then you'd stop, but you'd have a hard time getting your foot out because it's very sticky stuff.
I have lost a lot of shoes that way.
Yes.
We've got
not
some
down,
black corals.
Not down here, but you know. Oh, those look beautiful.
Yeah, they
Amanda,
do.
what are those?
Yeah, so those are black corals. Bathypathes is the genus. And neat. They can't live in sediment. They have to be attached to something firm. So just under there's probably a rock for them to be holding on to. Last year we tried to take a push core in an area that looked similar to this, and it was really, really dense clay. So for anybody who has clay soil, perhaps in their yards, and they try to dig into it, you know how it's firm and kind of sticky? that's my guess of what that lighter sediment feels like. And I don't know if that can be a good surface for the black coral to attach to or not, but it seems like whatever they're attaching to is working for them, whether it's a submerged rock or if it is that clay.
You guys are really making me want to go play in mud clay, deep sea sediments. It just sounds like fun. I bet it smells so weird, but it sounds like fun.
So there's a grenadier there, head up the slope.
Oh, there it goes. Oh, look, a little red jelly.
There's a benthichodon.
Yeah, a jelly.
Very nice.
That's amazing.
All right, I've got a pretty good question here from Leanne. I kind of want to know everybody else's opinion on this one as well. So she asks, I'm interested to know how what you see, discover during dive time changes you as a person. So what do you guys think? Do you think this experience changes you as a person or what you explore down here?
I think it makes, well, it's sort of hard to believe that some of these animals live down here. I mean, and they're colorful and bright and so varied. And it makes you think, what else don't we know? So if it changes your person, I think it makes you more curious. It's like, well, what is right around the corner? Makes you want to keep looking.
Yeah, for me, the first time I saw the deep sea was, well, in documentaries. At that time, I thought they were just absolutely bizarre. But when I went for my internship at the Monterey Bay Aquarium Research Institute as a summer intern, I went out to sea with the lab that I was interning with, and I saw the deep sea floor, and I couldn't believe that we were able to see something that was in an area so inhospitable to life. It kind of made me think of astronauts and the search for life in places that we can't, as humans, live. And here we were getting to explore a place where we can't live, and we know that there's life present. And so it was just a way to really get to see, like, whoa, there's this whole world that's living down here that's absolutely different from anything that I recognize. And so for me, how did it change my life? It actually directed me toward wanting to study these as my career. So now I'm hooked on understanding more about the deep sea and understanding how the animals that live there have evolved to be adapted to that, and also how they make their living down here. 'Cause there's not a lot of food. It's super cold. Many of these are slow growing and have all sorts of weird traits like the bioluminescence that Erica talked about. So yeah, it's just, it's so bizarre. It opens up my world.
Just to add to what Amanda said, is we know more about the surface of the moon than we do about the bottoms of the oceans. And this is the first time anyone's ever seen what we are all seeing right now in this spot. So that's pretty crazy.
It's true. We're sharing this discovery with everyone who's viewing right now.
So I would actually like to turn it around to the person who submitted the question, because now you're able to see the things that we're seeing. So how does it change you as a person? Does it
Is
make
that a shark?
A shark?
In the Argus cam.
I think it's a grenadier.
Grenadier,
Oh,
right.
I got excited.
No,
I'm sorry to
that's
interrupt.
the bubble cam, yeah. So, you know, I mean, for me, it's like both Amanda and Erica said, I start asking questions. How does this happen? How did this come about? What? How do these animals interact with their environment? But I ask those kinds of questions because I'm a scientist and I'm thrilled by the world around us and all the cool, weird stuff that plants and animals do. But, you know, you people watching at home can be having exactly the same